Hi, everybody. There's nobody here, but Amanda and I are still going to record it. We're doing week 4 review 2 session, and we're going to go over translation a little bit, practice problems, and point mutations.
Sounds good. So first, I'm going to give you guys a little bit of an overview of just translation as like a broad concept. So translation is the process of going from an mRNA sequence that you've made through transcription from your DNA. So you have this mRNA sequence and you're using that to make a protein. And translation uses 3 components. It is the ribosome, which is made up of the small subunit and the large subunit. And these are made up, the ribosome is like generally made up of protein and also some rRNA, ribosomal RNA. And there are also these tRNAs, these gold things right here, that bring in the amino acids that are used to make the protein. And then the mRNA, which dictates the sequence of amino acids to make up that protein. There are 3 steps of translation. The first one is initiation, start of translation. Then elongation, which is a lengthening of that polypeptide chain, the protein chain, by adding more and more amino acids to the end of it. And then termination, which is the end of translation, where you have the finished polypeptide, the finished protein primary structure leaving the ribosome. So like I just said, the sequence of nucleotides in the mRNA is what dictates translation and dictates the amino acids that are brought in to make up that polypeptide chain. Every set of 3 every set of 3 nucleotides is called a codon, and those codons, that like set of 3 nucleotides, is the reading frame. So every 3 nucleotides that makes up the codon is in that reading frame. If you disrupt the codons or the reading frame, you can cause some kind of mutation, which we'll go into in a little bit, that can change the amino acid sequence of the polypeptide. Okay, so quick overview of the ribosome. The ribosome has 2 subunits. There's the small subunit, which is this part down here, and the large subunit, which is this big part up here. And the ribosome is made up of both rRNA and proteins. The ribosome it's a type of structure called a ribozyme, which means that it has RNA that has a catalytic function. So this rRNA is a catalytic component of the ribosome, and therefore the ribosome is a structure called a ribozyme. It's a type of structure called a ribozyme. These 2 subunits, the small subunit and the large subunit, kind of sandwich. It comes on either side of an mRNA strand. It collapses on top, like on either side of that mRNA strand, it makes a sandwich. And the large subunit has 3 pockets called the E, P, and A sites. You guys don't need to know exactly what the E, P, and A stands for. But a good mnemonic that helps is the Environmental Protection Agency. That's one Laura taught me when I was a freshman, and that's stuck with me since. But those E, P, and A sites are all different positions in the ribosome where there may be a tRNA. Based on that positioning, the tRNA will be doing something different. So moving on to the next part of next thing that's involved in translation is the tRNA. The tRNA is a non-coding RNA, so it's not actually used to code for any proteins. mRNA is what codes for proteins. The tRNA is a non-coding RNA that's necessary for translation. It has something called an anticodon, which is these 3 nucleotides right here, these 3 bases. And the anticodon here pairs to the codon on the mRNA. We know about the base pairing, that A pairs with T and G pairs with C. So based on that, if, for example, here you have the GCC or GCG, this would, on the other side, on the mRNA strand, there'd be a C, G, and C that complementarily base pair to this. So these codons base pair to the codon on these anticodons, base pair to the codon on the mRNA, and each tRNA has a specific amino acid connected to it. So each tRNA has a specific anticodon, and that anticodon corresponds to a specific amino acid. There's one special tRNA that you have to know about. It's called formal methionine. This is special specifically in prokaryotes. It binds to the AUG the start methionine. You guys know how methionine is usually the start site, is at the start site. And that formal methionine tRNA, that's initial methionine tRNA, is the tRNA that binds to that initial AUG in the mRNA strand. So we can do this little question here. How many possible anticodons can a tRNA have? I think it's 4 times 4 to the 3. I don't know what that is. 16 times 4. 64? 64, I think. Yeah, that sounds right. 64. Okay. All right, so now we get into the 3 steps of translation. The first step is initiation, and we're going to talk here in detail about prokaryotic initiation. That's what you're mostly responsible for understanding. One key thing is that prokaryotic and eukaryotic initiation processes are different, right? They're fundamentally different in terms of how the ribosome attaches to the mRNA and how the process of translation starts. So in prokaryotes, the small ribosomal subunit, so this bottom subunit right here, binds to the mRNA first at what's called the Shine-Dalgarno sequence, or the ribosome binding site. Shine-Dalgarno sequence and the ribosome binding site mean the exact same thing. It's a specific sequence of nucleotides in the mRNA that the small subunit can recognize and bind to specifically. That first formal methionine, the tRNA that's carrying that formal methionine then binds to the start codon, the AUG of the mRNA that's on the mRNA. And then the large subunit then comes in and binds such that that formal methionine tRNA is in the P site, this middle site right here. You can actually see this is right after initiation has occurred. The formal methionine is sitting in the P site. You have the small subunit and the large subunit sandwiched around the mRNA. And it's at the formal methionine is at that AUG start codon. And then, yeah, so the Shine-Dalgarno sequence and the way that the small ribosomal subunit binds to the mRNA situates that AUG start codon in the right place, such that when the large subunit binds, the tRNA just is in that P site. Eukaryotes do this slightly differently. Basically, instead of it being, actually, Kira, do you want to say like 2 cents about that?
Eukaryotes do initiate translation a little bit differently. So with eukaryotes, instead of the small ribosomal subunit binding, eukaryotes, I'm pretty sure that the small subunit binds at the first methionine, and then the large subunit comes in. There's no, like, it just, no, sorry. Both of the subunits bind at the 5' UTR, I think at the 5' G cap specifically, and then they just, what's the word, like scroll, travel around along the mRNA sequence until they find the first start site. And that's how eukaryotes do it, which is different than prokaryotes.
Yeah, I think it's basically the in prokaryotes, the order is the small subunit and then the tRNA and then the large subunit.
Yeah.
In eukaryotes, the order is the small subunit finds the tRNA first, binds to the mRNA, scrolls a little bit until that tRNA reaches the AUG start codon, and then the large subunit comes in. So the order is a little different. Basically,
yeah.
For prokaryotes, it goes from bottom to top, basically the small subunit, then the tRNA, then the large, versus for eukaryotes, the small subunit and the tRNA meet outside of the mRNA, and then they attach the mRNA at the 5', scroll along until it gets here, and then again, the large subunit comes in last. Yeah. But yeah, this is a nice little picture of the initiation of translation in prokaryotes. Through the Shine-Dalgarno sequence, you can see that this is what this small subunit recognizes and binds to, and it binds to this in such a way that this AUG site is in exactly the right place for this methionine to come in and bind, so that when the large subunit comes in, it associates with the small subunit, that formylmethionine tRNA is in that P site. All right, so next up is elongation. So this is, we've started the process of translation. We had that formylmethionine. We have this whole thing right here, and now we need to add more amino acids to this to make a big long chain of amino acids. So the next tRNA comes in and binds at the A site, right? Based on the codon and anticodon pairing between the codon on the mRNA and the anticodon on the tRNA. That binding occurs at the A site. The polypeptide chain that's held in the P site, whatever is there, at this point, it's just the formylmethionine, makes a peptide bond with the amino acid held by the tRNA in the A site. Then that polypeptide chain is like, that lets go of the tRNA in the P site, right? So now it's just held on, excuse me, it's just held on by the tRNA in the A site, and the entire ribosome shifts down one slot. So the A, what's in the A goes into the P, what's in the P goes into the E, and then what's in the E just pops off. And this is repeated over and over. There's a nice little diagram right here. So you have this shows the very first, the methionine is right, formylmethionine is right here. Then you have the next amino acid coming in that slots into the A site. There's a bond that's formed right here between the methionine and the next amino acid. So that lets go of the tRNA in the P site. And now that's all just held by the tRNA in the A site. And then the entire ribosome ratchets over one. The empty tRNA now is in the E site. The full tRNA is now moved from the A site to the P site, and whatever's in the E site automatically just pops off. That empty tRNA just pops off. And now you have a chain of two. Now you can imagine if you go back to here and add another amino acid coming in, that chain becomes 3, 4, 5, and so on until you get hundreds of amino acids chain, like in a big long string. Okay, now the last step is termination. After the last pair of tRNA amino acid comes in and binds to the codon on the mRNA, you have your full polypeptide chain. The next thing they'll show up is a stop codon, right? So before that moves into the ENP sites, the termination factor, which is a protein and not a tRNA. This is not an empty tRNA. Don't think of it as an empty tRNA. It's a totally different protein called a termination factor. That comes in and binds to the stop codon. So now that's sitting in the A site. You have your whole chain, or sorry, that's sitting in the A site. You have your whole chain in the P site, but nothing has moved into the E site yet. A good question is how many termination factors are there? The thing to think about here is the fact that there are 3 stop codons. You good animal, you ate grass, you are awesome. So it's UGA, UAG, UAA. Those are 3 stop codons. And because this protein has to be specific to stop codons, there has to be 3 different proteins that bind to those 3 stop codons. So 3 types of 3 termination factor. Like the termination factor can bind to 3 separate stop codons. So there have to be 3 separate termination factors that are specific to each of those stop codons. Once a termination factor binds, that termination factor causes the cleavage of the bond between the P site tRNA and the polypeptide chain. So now that tRNA is still in the P site, but now it has let go of the chain of amino acids. That chain of amino acids is now free. That can fold, it can do whatever it needs to do. You have your full primary structure of protein. You have your polypeptide chain. It has been made. Translation, for all intents and purposes, is done. Then the entire thing kind of falls apart. The small and large ribosomal subunits come apart. That empty tRNA leaves, the termination factor leaves, and the mRNA that was being used by the ribosome also leaves. That mRNA can then be recycled, and translation can happen again with that mRNA as many times as possible before it eventually gets broken down. So this is like an image of like release. You can see the last tRNA is here. You have the stop codon. It's U. How do I get this way? You good animal. You ate grass. You are like you good animal. You ate grass. You are awesome. The release factor, this protein, comes in here. It cuts off the polypeptide chain, and the entire thing comes apart. Key thing to remember here: this is a protein and not another tRNA. All right. So speaking of translation and stuff like that, an important part of that is reading codons and translate. Like if you are given a DNA sequence, you should be able to translate that or transcribe that into an RNA sequence, an mRNA sequence, by finding the opposite kind of corresponding letter. So G's bind with C's, A's bind with U's, T's bind with A's, if you're going from RNA to DNA to RNA. Now, if you're going from RNA to protein, you need to be able to translate each one of those codons into an amino acid. So let's say your codon is UAC, right? You find the first, or let's actually do the example, it's right here. If your codon is CGA, find the first nucleotide on the left, right? That's C. You scroll over until you see where the second nucleotide is, that's G. So C and G, so it's in here. And then you look for this, the last letter in the codon right here. You look for A, you see GA right there, that corresponds with arginine. Keep in mind that sometimes that just the fact that it's in this box doesn't necessarily mean that it's a specific amino acid. In this case, anything that starts with CG is an arginine. For example, with the AAs, AAU and AAC are, what's that? Asparagine?
Yeah.
But AAA and AAG are lysine. So make sure you're very careful and thorough when you're using these codon charts. You will have to use these codon charts to go from mRNA to protein on problem sets and exams and things like that. All right, so that's how you read a codon chart.
Really quickly, we're going to go over some practice problems, but nobody's here, so we're just going to tell you the answers basically and work through how to do them. So we're given this mRNA strand right here, 5' AUG UUA, and then so forth until the 3'. And the question is asking how many amino acids does this mRNA code for? What is the polypeptide sequence? So from the first question, we're going to figure out how many amino acids this mRNA codes for. And so we know we can figure out how many amino acids a strand codes for by figuring out how many sets of three nucleotides there are, minus the stop codons. So in this case, we have 1 AUG, 2 UUA, 3 CGU, 4 GCU, 5 AUC, 6 UAU, and UAA is a stop codon. So again, just to reiterate, a stop codon does not code for an amino acid. It codes for a release factor, which is a completely separate protein. So although that UAA is a codon, it does not code for an amino acid. So in this case, there are only 6 codons that this mRNA codes for. So we got that by counting up how many sets of 3 nucleotides there were, minus the stop codons. And so we can figure out what the polypeptide sequence is by looking at the codon chart. So with AUG, we know that that's going to be methionine. UUA is going to be leucine. Yeah, UA is going to be leucine, and I can just read off the rest. CGU is going to be arginine. GCU is going to be alanine. Yeah, GCU, alanine. And then AUC, isoleucine. And then the last one is UAU, which is going to be tyrosine. And then that UAA again is going to code for a stop codon, non-amino acid, so we don't count it. But that's going to be the polypeptide sequence. The first amino acid, methionine, is going to be at the N terminus, right? So we discussed N terminus and C terminus in a previous, like before the last exam, the N terminus basically designates the free amino group, which designates the beginning of an amino acid sequence. And then the C terminus designates the free carboxyl group of an amino acid sequence, which also basically means the end of an amino acid sequence. So that's the polypeptide sequence. And if you were asked to do this from the coding strand of the DNA, it's really easy. So if we remember correctly, the coding strand is not going to be the strand that is being used during transcription. So it's going to have the exact same sequence with the exception of U's and T's as the mRNA that's being transcribed. So if we see here, the coding strand looks the exact same as the mRNA. Oh, JK, I did not read this question closely enough.
It's just the AUG.
Okay. My bad, guys. So if you were asked that question, that's how you could do it, but we're going to go through. So we can figure out the mRNA by basically just substituting the T's for U's in this case. And once we have the mRNA strand, will allow us to figure out the amino acid sequence. So from the coding strand, it's going to be 5' AUG, which is methionine, CGC, which is arginine. Yeah. TTA, sorry. Since we have TTs in DNA, we're going to switch them out for uracil. So it's going to be UUA, which codes for leucine, right? Yeah. And then we have CCT, but that T is going to become a U. So it's going to be CCU, and it's now going to become proline during translation. And then we have AGA, which is going to become arginine. Yeah. And then after that, we have ACC, which is threonine. And then lastly, we have TAG, which is going to resemble, or it's going to be basically UAG in the coding, or sorry, in the mRNA. And UAG is a stop codon, so that will not code for an amino acid. It's just going to code for a release factor. So that's the end of the polypeptide. In this case, we have 1, 2, 3, 4, 5, 6. We have 7 codons, but we only have 6 amino acids that the coding strand of DNA actually codes for a protein. Okay, next question really quickly. If you were asked to produce the amino acid sequence from a template strand of DNA, oh boy.
Oh, am I back?
My bad.
Am I back?
Yeah, it was me. My
Okay.
computer like disconnected from it, Jerome.
Excellent. How many codons are in the coding sequence of this gene?
Okay, so if you were asked to produce the amino acid sequence from that template strand of DNA, you could do so by finding the complementary base pairs of each nucleotide, while keeping in mind the anti-parallel nature, so 5' and 3' ends. So in this case, if you were to transcribe that mRNA that would be produced from that template strand, it would be 5' going from left to right, 5' A AUG, which is methionine, and then you would have ACC, which is threonine. After that, you would have GCG, which codes for alanine. After that, you would have GGU, getting lost, which is glycine, I think. Yeah. And then you have UUU, which is phenylalanine. And then you'd have GGG, which is glycine again. And you would have UAA, which is a stop codon, so it would end translation entirely. But if you were asked to produce the amino acid sequence from a template strand of DNA, you would find the complementary base pairs, or you would create the mRNA using complementary base pairing. And then from there, you would just use the codon chart to figure out what each codon means. Not that bad.
Nice.
Okay, we're gonna go over point mutations too. So most often in our DNA, we have a bunch of mutations, or that happens whenever it's dividing. It's just a consequence that happens. But in this case, we're going to be talking about single nucleotide point mutations. So that's involving one nucleotide in the reading frame. And that can have three effects. So it can either be a silent, a missense, or it can either be classified as a silent, a missense, or a nonsense mutation. So silent mutations are when you alter the DNA sequence in some way, but the end product is an amino acid that is still the same as the original one. So if we look at that chart really quickly, yeah, over to the right, the original sequence was TAC, but we changed that to TAT. So during translation, TAC would have coded for a tyrosine. And in this case, with the TAT, the outcome is still a tyrosine. So the primary structure does change, which is very important to note, but overall, it's probably not going to change tertiary or quaternary structure just because it's the same amino acid. So it's going to have the same chemistry and most likely the same shape and function. Missense mutation are when one amino acid is swapped out for another. So that happens when you change the DNA so that when translation comes around, it's going to be the codon like in question is going to code for a completely different amino acid. And this can have a small effect or it can have a really big effect, but most likely it's not going to be that bad just because it's one amino acid. So if we look at the chart again, the original amino acid sequence was TAC, and in this case, we changed it to AAC, that single point mutation, so the T was changed to an A, and this actually codes for an asparagine instead of a tyrosine, so it's going to be a missense mutation, not a silent mutation, because you're swapping out one amino acid for another. And then one of the second to last type of mutation that we're going to talk about are going to be nonsense mutations. So this is when the DNA is changed so that the stop or the codon that was originally going to be coded for is now a stop codon. So this ends translation and it can be prematurely, or it will most likely be prematurely because you're ending translation before it was supposed to end, I guess. So if we look at that chart really quickly, we have TAC, but in this case, we change it to TAG, and that's actually going to code for UAG, which is a stop codon. So this ends up with translation ending, and this can have really drastic effects because if you change the DNA so that you're like halfway through translation and you come across a stop codon, you're going to be missing that other half of the protein or the other like half of the amino acid sequence, and that's going to have drastic effects. So a lot of the times this is a very harmful mutation. And in these cases, you can have mutations or point mutations in other parts of the gene. So you can have them in the 5' UTR, in introns, stuff like that. But because we're talking about translation and point mutations, we're only talking about what's in the reading frame. So that's only referring to the exons in this case of the gene. It's not going to be talking about the 5' UTR, the 3' UTR, basically anything that's not translated. So that's important to note.
Yeah.
Next one, please.
One thing. Oh, yeah. One thing I want to add really quickly is that I really want to stress what Kira said about like what the effect is on the protein. Like if you have a silent mutation, the reason that I don't know why it says tyrosine, it's like a misspelling of tyrosine.
Did not
but one of the reasons that we don't go right there. One of the reasons that you have this whole thing, there's a lot of redundancy when it comes to translation in terms of like ACU, ACC, ACA, and ACG all code for the same amino acids. You might be thinking, what is the point of that? Why not just have ACU do that? And the reason is that makes it easier to have silent mutations. If you have a point mutation right here that changes this from a U to a C, you're not changing the amino acid. You're keeping the primary structure the same. It's not gonna have any effect on the actual protein, the way the protein folds, anything like that. The string of amino acids, that primary structure stays the same. So secondary and tertiary structures would also stay the same. When we get into missense and nonsense, nonsense is probably really bad because you're just cutting off a part of the protein. If that happens towards the end of your mRNA strand, towards the end of your primary structure, you might be able to get away with a protein that's like kind of able to do something. But if it happens like halfway through your protein, that protein is shot, basically. There's nothing that you can do with like a half a protein. A missense mutation where you swap from one amino acid to the other, in this case, a tyrosine to asparagine. Think about what you guys have learned about the amino acids in their chemistry, right? So you guys know that certain amino acids are non-polar, certain amino acids are polar, certain amino acids are charged, certain amino acids are positively, negatively charged, right? So if you're swapping out, if your missense mutation results in two amino acids that are very similar being swapped out, it will change the primary structure of your protein. It will change the sequence of amino acids that make up your protein. But if you're swapping out a small non-polar amino acid for another small non-polar amino acid, you might be able to get away with it in terms of what that amino acid does in the context of the protein and the resulting protein. So you might get a faulty protein that's harmful, you might not. It really depends on the chemistry of the specific amino acids that you're swapping out. For nonsense mutation, you're probably screwed. For silent mutation, you're fine. Does that make sense? There's nobody here to answer that. Kira, does that make sense?
Makes sense, Aman. Thank you for explaining that.
Nice.
Okay. The last type of mutation that we're going to discuss is called a frame shift mutation, and that's when a nucleotide is added or removed from the sequence and the reading frame changes. So you can imagine why we call it a frame shift mutation. When if the reading frame changes, then all of the other codons downstream are going to be slightly out of place. So it's going to result in a massive missense kind of effect where all of the amino acids, all of the amino acids downstream are going to be swapped for another one just because it's going to move like one letter over, one nucleotide over, one nucleotide before, just because you're adding or deleting a nucleotide. So just to reiterate, because I don't think that was very clear, but in this case, I am adding or deleting a nucleotide, and the result of that is that the reading frame changes because the codons are not being read in the same 3 pairs that they were being read originally. So in this case, we're only talking about point mutations, and that's like a single addition or deletion. But if you add or remove 3 nucleotides, it's not considered a frame shift mutation just because all of the nucleotides downstream or all of the codons downstream of that 3 nucleotides, whether they're added or deleted, are going to be the same. So the frame isn't going to shift. All of the amino acids downstream of that are going to be the exact same. So it's not a frame shift just because everything is still in frame after. But if you do have an addition or deletion of anything not in a multiple of 3 nucleotides, then it is a frame shift mutation, and that has drastic consequences because if you think about it, everything is out of place now, and the protein will most likely not look anything like it was before because all of the amino acids downstream of that frame shift mutation or of that mutation are going to be different. So that's going to have massive impacts on the chemistry of the protein because it's not going to be able to do what it was originally. And it could result in either a longer or shorter protein depending on where the stop codon, or like if the stop codon is out of place. So basically, this is probably the most harmful mutation that we've talked about, and it has very severe consequences because the protein isn't going to look anything like it was supposed to. So just to reiterate, unless if you want to do it, doesn't
Sorry,
matter.
I realized that I started talking, but I realized I was muted.
That's fine.
So just like a big overview of everything we've talked about. Translation occurs in 3 steps. There's initiation, elongation, and termination. And initiation, the main thing that or for the translation steps that you really have to think about are the prokaryotic stuff. Just know that eukaryotic is different. In prokaryotic translation, you have the small ribosomal subunit binding to the Shine del Garno, or the ribosome binding site in the mRNA. Then formylmethionine comes in and binds to the AUG. Then the large subunit comes in and binds such that that formylmethionine tRNA is sitting in the P site. We then move to translation elongation, where new tRNAs carrying amino acids enter the A site and bond to the anticodon on the tRNA bonds to the codon on the mRNA. Then you have the amino acid of the A site tRNA bonding to the P site amino acid, P site tRNA's amino acid, and the P site tRNA lets go of the amino acid chain. So now it's all attached to the A site tRNA. The ribosome shifts so that former A site tRNA is now sitting in the P site. The old tRNA is now in the E site and can leave, and a new tRNA can come into the empty A site. When we get to termination, you get to when there's a stop codon, a protein called a termination factor binds instead of any tRNA. You don't get any peptide bond formation, but instead the entire polypeptide chain that you've made up until that point is released from the tRNA that's sitting in the A site. So that polypeptide can go do whatever it has to do, and the ribosome disassembles. The tRNAs come off, the termination factor comes off, and the small and large subunit come off. And the mRNA can possibly be read again by a different ribosome, and same ribosome and same one, whatever. It can be read again if the opportunity presents itself. And then we also talked about mutations. Point mutations change one nucleotide to another. You can have a silent mutation, a missense mutation, or a nonsense mutation. Frame shift mutations have to do with insertions or deletions of a nucleotide, and they have the ability to change all of the amino acids after that by changing the reading frame. And that changes like which codons are considered, like it changes how the codons are read. So you can screw up all the amino acids at and after the point of the mutation. Yay, that's the big picture.
That's all we have for you guys. Nobody's here, but if you do see this recording and you have any questions, please feel free to email me or Aman. Otherwise, have a nice night.